presenting this on behalf of Serafina Pacilio, that is a talented PhD student that is now in our lab, and she works under the supervision of Professor Cenacchi, and I thank you, Professor Garan. So, we are talking of this disorder that is a very rare muscular dystrophy, autosomal dominant, with a juvenile or adult onset, with a variable degree of pelvic weakness. And this disorder has been characterized largely by the group of Giovanna Cenacchi and other colleagues. What we have is that we have a mutation, autosomal dominant, in this protein that is transportin 3, that is a nuclear import receptor for serine arginine-rich proteins, and all the patients have different variants, but that lead to the same effect. I don't know how this works here. That lead to the same effect where you have a longer protein with a probably gain of function effect. And in the muscle biopsy of the patient, we have a fiber atrophy with some internalization of nuclei, we have autophagy, we have myofibrillary disarray, and you have these aggregates of SRSF1. So, we don't know exactly what is the function of this protein, but as this is a nuclear import factor that is very important for the cargo protein, in this C-terminal domain of the protein, there is an important role that protects this patient from the integration of HIV. So, unfortunately, they have a muscle dystrophy, but they are protected from these disorders. There are different working hypotheses about this condition. So we have an autophagy accumulation that you can see here as demonstrated by the accumulation of PC52. We have an SRSF1 dysregulation that probably provoke an altered myophagy. And to demonstrate this, Serafina and the group of Cenacchi did a lot of studies, including investigation of transportin 3 and SRF during myogenesis. They characterized the muscle biopsy. They do silencing by CRISPR-Cas9. They created zebrafish model. And the objective of Serafina project, thanks to this collaboration with Italian and the French group, is to study proliferation and differentiation on different cellular models, comparing patient cell with wild-type fibroblasts. So the methods are here. She performed in Italy two different studies, one characterizing A2D cell culture model, a standard model, and the second one, they used this very fancy 3D collagen scaffold in order to evidence if there are differences in the two systems that allow the characterization of the myoblast of the patient, where in the system 3D we have a functional biomaterial that allow a better study of the interaction of the cell with the extracellular matrix. Here you have the representation on how these gel are done. So you have a 3D printer with bioink that allow to create these scaffolds. And here you have the bioprinting process in this video. So when we studied, she studied the myogenesis, she looked at different factors of proliferation and differentiation, and already defined alteration in MIF, in myogenin, and then the characterization of the cargos were also altered. Here you have the results. You can see that in the patient's myoblasts compared to the control, we have a late expression of MIF5 that probably means a prolonged attempt for the cell differentiation. We have an increased and prolonged myogenin that means a continuous stimulation of these cells in order to go into a full differentiation. And then when they checked all these factors, what they found is there was an increased transportin 3 expression that compensates probably the dysregulated activity leading to alteration in cargo levels. The markers of autophagy were also altered in both the 2D and 3D models that it's really corresponding to what was found in the muscular biopsy of this patient. And by elegant electron microscopy studies, as you can see here in the control where we have a right alignment of the filaments, in contrast, in the patient cell we have a completed disarrayed filament structure. So, in order to conclude the first part, what they demonstrate is that we have an altered gene and protein expression, the transportin 3. We have a similar result in both T2 and 3D models, with an enhanced scaffold tendency of mimicking what we found in the muscle, in the muscle of the patient. We also showed that there is a delayed expression of MIF5 and elevated levels of atrogene and P62 that are related to this autophagy dysregulation. When Serafina came to France, so she was working on these 3D models using pillars that has been developed by Nathalie Didier and a PhD student, Gisela Banos. And you can see here that what we do, we seed the myoblast of the patient on this plate, where there are two pillars distantiated by 3 millimeters length. And after a seeding during 2 days, what we have an association of the myoblast to the different pillars, and from 7 to 10 days, we can see a contraction of the pillars, hopefully. So what was done an analysis of the myoblast immortalized and the control line. And as you can see here in the picture, we have already at 7 days an altered positioning of the nuclei that stay inside the cell in a central position compared to the control myoblast where the myonuclei are peripheral. What we also found at 10 days, that we have an abnormal branching of the myoblast, as you can see here, and also you have this accumulation of actinin inside the muscle fibers. And here, I hope that the video works. There is a very different phenotype in the patient myoblast compared to the control, where if you look at the lines, you can see that there is an hypercontractility phenotype with a peak that lately go down in the myoblast of the patient compared to the control. And other studies with the tetanus contractile force show that we also have a very high peak that decreases successively compared to the control. So these are very interesting but preliminary results that show that we have a myofibers irregularities in both models, both the 3D scaffold performed in Italy and the one with pillars that we studied in France. We confirm that the cell functionality is altered, especially for contractility and electrical stimulation. And we have this twitch contracture and tetanic contracture that is in favor of a gain of function mechanism of the disorder. And the fluctuating contractile force observed in the patient cell suggests a potential correlation with an altered motor process. Of course, we need to confirm this. Serafina probably will stay with us for more time, and also she will work on a mRNA. She will perform the adaptability and repeatability study of the system in order to create novel in vitro models, and then the next steps will be to perform some drug repurposing studies and drug testing in order to find a possible therapy for this very rare disorder. So I would like to thank you for the attention. Of course, this is the work of Serafina, the group of Professor Cenacchi in Bologna that co-tutor with Professor Focarete and Serafina, and of course the group of Nathalie Didier that provided this model. Thank you very much.